Good evening and welcome to this PSC Partners Roadmap 101 meeting. The PSC Research Landscape, Now and Into the Future, 5 Concepts in PSC. My name is Mary Villas and I'm the president of PSC Partners Seeking a Cure Canada. It is so exciting that so many have joined us this August evening. This meeting will start with a general session about PSC research, followed by moderated breakout rooms where you can ask your questions. Next slide, please. Before we get started with this Roadmap 101 meeting, I'd like to briefly introduce what the Roadmap Initiative is all about. Roadmap is one outcome of work that has been ongoing over the past few years. This includes the receipt of the CZI Rare as One grant. A primary goal of this grant is to leverage the power of patients to accelerate PSC research, including the development of a patient-organized collaborative research network. The October 2020 PFDD Forum on PSC with the FDA, and the PSC Partners Patient Registry, which currently has over 1,900 participants from around the world. Together, this work has led to expanding the vision of the role of PSC Partners and the PSC patient community in research and drug development. Next slide, please. With that bit of context, let's revisit the goals of Roadmap, which are accelerating progress to find therapies and a cure by 1, educating the patient community on the research landscape, 2, broadening the researcher base and strengthening multi-stakeholder communication, and 3, creating opportunities for researchers and clinicians to engage in meaningful discussions with an informed patient community. Next slide, please. Practically, what does this mean? Hopefully, you have seen this roadmap graphic on social media or in newsletters. To the right, you can see the 3 main stakeholder groups, and on the left shows the 4 topic areas as steps. We're currently on topic 2, research. For each of these topics, we're hosting a series of meetings with 2 or more Zoom meetings for each topic. The 101 sessions, like tonight, are community-focused and educational, and the 201 and up sessions are multi-stakeholder discussions. Next slide, please. Thank you again for joining us, and again, welcome. Now I will pass the Zoom mic to Ricky Safer, founder and CEO of PSC Partners Seeking a Cure.
Hi, thank you, Mary, and thank you to all of tonight's attendees who are showing up in great numbers. We have a wonderful program tonight. This is the first of 3 research webinars. Our goal tonight is to educate our patient caregiver community on 5 central concepts in PSC, to give us the background to discuss these concepts with the medical community in our future research webinars. Here's today's agenda. Our keynote speaker, Dr. Heather Francis, a member of our Scientific Medical Advisory Committee, will give an engaging introduction to these 5 concepts. After her presentation, Dr. Ruth Ann Pye, Director of Research Strategy at PSC Partners, and Joanne Hatchett, Medical Science Liaison at PSC Partners, will give a brief introduction to our breakout rooms tonight. Now, I'm excited to introduce Dr. Heather Francis, Professor of Medicine, Indiana University School of Medicine. Her primary focus of research is PSC. Dr. Francis receives NIH and VA funding for her research, and she is currently serving as the chair of the Hepatobiliary Pathophysiology NIH Studies Section. Dr. Francis became involved with PSC Partners in 2013 when she received a grant from our organization, and she's been a loyal supporter since then. Thank you, Dr. Francis, for your presentation tonight. Please take it away.
Hi, thank you, Rick, for that introduction. And I just want to say good evening to all of the PSC Partners people that I see online. I haven't seen you guys in way too long, almost 1.5 years now. And I'm hoping that we get to see each other very soon in person. And I'm very honored to be here tonight to be able to talk about these basic concepts with the group before we go into the breakout sections. So next slide. So the overview here that I'll give is I'm going to talk a little bit about the effects that PSC has on the liver, and then some of these other important topics, including bile acids, genes, genetics, the microbiome and the gut-liver axis, and of course the role that immune cells also play during PSC. And I really want to emphasize that a lot of times when we're thinking about how our bodies are functioning, a lot of people don't really think about the liver. It's a very silent organ, and you don't feel anything like a heartbeat. And so we forget, I think, sometimes actually how hard our liver is working for us every single day. And most of us, unfortunately, probably are not always super kind to our liver. So we for sure want to raise our hats to all of the proper liver functions that we're appreciative of every day. Okay, next slide. So what are some basic functions of the liver? And there are actually over 500 functions the liver provides for us every day. But some of the really important ones are include immunity against infection, so keeping us clean from all of the harmful pathogens, regulating blood clots. This is also where we're going to get all of our proteins and cholesterol. And really important, the liver provides a mechanism to clear the body and the blood of all the drugs, chemical, alcohol, even damaging foods that you can eat throughout the day or over your lifetime. One of the other important functions of the liver is to excrete waste through bile, and we're going to talk a lot about bile as we go through the presentation. And then another one is to convert all of that excess glucose that we consume into starch for storage. And then again, excreting bile for fat digestion. Sometimes we like to look at the liver a little bit like the garbage disposal, because basically you can put just about anything you want into your body, and the liver, if it's functioning properly, will be able to filter through, save the nutrients that you need to survive and to promote good living and proper health, and then get rid of all of the toxins and the negative waste that you definitely don't need. And really the liver is always working overtime. So if you're spending time having a lot of fun, maybe too much fun, then your liver is definitely providing the functions it needs to continue with proper homeostasis and the rest of your organs also functioning properly. Next slide. So what about the impact of liver disease? We know that almost 5 million adults have been diagnosed with liver disease. There are about 14,000 people that are awaiting liver transplant. And out of 1 million people, 27 will receive a new liver. And these are stats from 2019. And in 2019, nearly 40,000 liver transplants were performed. These include both donor liver, but also live donor liver transplantations, which are becoming more frequent over the last decade. And really, when we look at liver damage, and this is just really a general scheme of this, so you have your healthy liver and you're able to function, everything is proper, you're able to break down harmful drugs or toxins. And then if you develop liver disease, then you start with maybe inflammation, scar tissue can build up within the liver. This then can become a fibrotic liver, which can progress into a cirrhotic liver. And this is extensive scarring, and this can actually block the blood flow through your liver. And this is a term we call cirrhosis, and can be caused by different causes. People usually associate cirrhosis only with alcohol, but that's not always the case. And then of course, liver cancer can be an end stage of the progression of liver damage. Next slide. But we need to talk about the anatomy just a little bit before we go into the next slides. And so we know that the hepatocytes make up about 97% of your liver, and they're also very dynamic cells as well in regulating homeostasis. And when I say homeostasis, I mean the balance of healthy versus not healthy, or good versus bad. And you always want to maintain a sort of a stable plane here. But the cell type that we're really interested in when we talk about PSC are cholangiocytes. And so if you look at the top here, and you can see the intrahepatic bile ductules, and this is where you're going to find the cholangiocytes, which are the cells that line these bile ductules. And if you look on the bottom, I put this tree up here because a lot of times we talk about cholestasis or PSC in terms of the biliary tree. And really, when you look at a cholangiogram of the liver, you can actually see that this has branches and it looks very much like a tree. And during disease states, you can either have loss of some of these branches, or you can have an overreaction of some of these branches where you have too many, and you have what we would call a very proliferative state. And then on the right, this is a drawing I always like to refer to. So we have the hepatocytes out surrounding the ductule that's lined by the cholangiocytes. And of course, this is going to lead into the common hepatic duct, where the bile will flow and out, be stored in your gallbladder or be removed. Next slide. So what are some of the effects about PSC on the liver? Well, one of them is bile duct damage. So the bile ducts can become blocked. So when you think about bile ducts, you can think about this in terms of a garden hose or any kind of tubing where something is flowing freely through it. And when it gets obstructed, then everything backs up onto the other end. And this is exactly what happens in bile ducts when they become inflamed or there is any scar tissue or fibrosis. And so basically the bile will accumulate in your liver, and this will gradually damage these liver cells, including hepatocytes. Cholangiocytes are typically very resistant to the toxic bile acids that are flowing through there. However, if too much bile builds up, then of course they can become more prone to damage. And bile is just that fluid that's made and released by your liver and stored in the gallbladder. And of course, this helps with your digestion and breaks down your fatty acids, and a very, very important molecule, but can be really damaging if you have any kind of a kink in your bile duct or any kind of inflammation or scar tissue. Next slide. Another part of PSC that can be quite damaging is the inflammation, scarring, and fibrosis. So inflammation, this is just a reaction that happens when your cells in the liver are basically being attacked by other foreign microbe substances. And then fibrosis is the formation of abnormal scar tissue in the liver. And this can actually cause strictures, which is the narrowing of the bile duct. So as the water hose is getting kinked, then you might have a narrowing within the bile duct, and this will actually increase the damage to your liver as well. And if you look down here, I don't know how well you guys can see this, but this is a little tiff on the brain liver axis, because everything is controlled by the brain basically, and the liver here is incredibly upset because the brain does not have things under control, and he's down here, he's babysitting the gallbladder, he's busting the bile ducts for the body. The brain is just sitting in the ivory tower thinking about philosophy, and the liver is saying, watch the input, don't make me angry. And then of course they're like, that wasn't already very angry. The liver is just very passionate about their work, right? Liver is just trying to keep everybody happy and healthy. Because when the liver has balance, then this is going to affect the other organs in your body as well. Next slide. So one of the last ones, when we talk about PSC direct effects on the liver, is the cholestasis and cirrhosis. So again, we talk about cholestasis in terms of buildup of bile. So the kink in the hose has been sort of irreparable. All the bile is backing up. And basically cholestasis, the literal meaning here, means standing bile. And when you have this toxic level of bile and bile acids that have infiltrated your liver, then this can also lead to the cirrhosis due to biliary tree damage. Not alcohol-induced liver cirrhosis. This is very different. This is a very rare form of liver cirrhosis. And again, I like to show the tree here because we have this balance between having maybe too many bile ducts, too many proliferating cells versus the cells that are not functioning at all. Next slide. Okay, so what about bile acids? Let's look at these a little bit more closely. So these are produced by hepatocytes and they're secreted into bile. And as they're secreted, they're modified by cholangiocytes in the bile duct. Remember, I told you that cholangiocytes are resistant to the toxicity of the bile acids, and they aid in the digestion and breakdown of your fats. And then there's a sort of a conversion of bile acids. You have primary bile acids that come from cholesterol, and then they're modified by cholangiocytes into these secondary bile acids, and also through the gut bacteria. And then we can also have alteration of bile acid composition itself. So if we look at bile acids, I didn't want to really go into this too deeply. There are so many different kinds, and there has to be a nice balance of these bile acids. And when we have bile acid toxicity or an alteration in their composition, this can also contribute to liver disease. And if you look down here at the bottom, we have what's called enterohepatic circulation. This is just the circulation of bile acids within the liver. So basically hepatocytes say, hey, bile acids, get out. They think that they're done. The next thing they know, bile acids are right back, coming right back behind them. And this is called intrahepatic recirculation, where we have recycled bile acids by hepatocytes. Next slide. If we look at this a little bit closer, so I have intrahepatic circulation and also systemic circulation. So again, we have the cholangiocytes that are protected from your toxic bile acids. We have a compound called Glycocalyx, which is like a gelatin coating. If you think about a fish, that's when you pick up a fish, it's very slimy and it slips out of your hands. That's Glycocalyx. And the same thing is also on cholangiocytes to protect them from bile acids. And one of the components that I wanted to mention tonight is this FUT2 that regulates the sugar formation of glycoproteins and glycolipids. And what they found is that in populations of patients with PSC or Crohn's, that there's inactive FUT2 in some variants. And what happens basically is the glycocalyx can't protect the cholangiocytes anymore. And so in certain subsets of patients, this glycocalyx may not be working and the cholangiocytes are now prone to damage. Another player here is TGR5, which is just a G protein coupled receptor. But what its role is to hold on to junctions within the gut. And we'll talk about this more when we talk about leaky gut in a few minutes. But basically, if you have a dysfunctional regulation of TGR5, then you might also have leaky gut that can contribute to your PSC prognosis. Next slide. So what about bile acid therapies? I'm only going to touch on this. So we have UDCA or ursodeoxycholic acid, and we have obeticholic acid or OCA. So your UDCA has been around for a while. This is a drug that's also used for primary biliary cholangitis patients. This is also used in combination with antibiotics. It can decrease bile acid toxicity, and it can even change your bile acid species composition. And this has been shown in different models. You might also get some relief from the pruritus or the chronic itching. The bad news is that not all patients, not all PSC patients will respond to therapy, to UDCA therapy. And there have been other studies that have shown that depending on the dose, it might actually make things worse. Another derivative of UDCA is called nor-UDCA that's had a better effect, and it works in the same manner. And the newer one is obeticholic acid or OCA. This is an FXR agonist, which is a receptor that shuttles bile acids. And basically, we can reduce the serum alkaline phosphatase levels in PSC patients with OCA. There have been some side effects though, and one of them happens to be increased pruritus. These studies are still ongoing, clinical trials, and OCA is also being evaluated in non-alcoholic fatty liver disease and NASH and other modalities. The thing you have to remember about bile acid therapies is anytime you're altering your bile acid composition, you might have other consequences from that. Next slide. So what about the genes and genetics? There are about 16 high-risk genes that have been reported. HLA, or the human leukocyte antigen genes, are one of them. And these also present as MHC2, which is a molecule that basically senses foreign antigens and proteins. And in patients with PSC, there's a genetic tendency mainly associated with HLA, MHC class 2 chromosome 6p21. And so these are some studies that are ongoing. And there are other variants that are also associated with IBD and immune genes. But unfortunately, it's not so simple to just find the gene that causes PSC, since we know that this can also arise from other factors and not just genetics. Next slide. All right, what about the microbiome? So the definition of microbiome is the community of the microorganisms that live together in a particular habitat. And our human microbiota has a lot of bacteria, archaea, fungi, protists, and viruses. And it can sound a little creepy to know that all of these creatures are living within us, but this also happens on your skin, mammary glands, your lungs, your eyes, and the biliary tract and the GI tract, of course, as well. And I mean, I don't know about anybody else, but I totally agree that we have to support bacteria because it really is the only culture some people do have. Next slide. So let's talk more specifically about the gut microbiota. So this is the totality of all the microorganisms that are present in your GI tract. These are going to be really important in nutrient, mineral absorption, synthesis of proteins, enzymes, vitamins, amino acids, and also the production of your short chain fatty acids. So why is this such a big deal? Basically, when you have an imbalance or what we call dysbiosis, which is just you want to have a very diverse microbiome. You want to have a lot of different players in there that have different roles. And when you have a switch or a change in your microbiome due to disease, due to something you ate, due to a genetic factor, due to pollutions, then you have this dysbiosis, and this can cause a dysregulation in your gut homeostasis or in the balance of how your gut is functioning. And again, a lot of factors can play into this: diet, even where you live. So if you spent your whole life living in Greece, your microbiome will be set in a certain way. And if you move to America and you start eating in a different way, you can actually change your microbiome, and it can induce problems, not necessarily disease, but intestinal issues and other things that can affect even your liver and other organs because you've changed your microbiota. Next slide. So that leads me to talk about a little bit about IBD and PSC. So we know that about 70% of patients with PSC also have irritable bowel disease. It's usually ulcerative colitis. And then really only about 5% of patients with ulcerative colitis will develop PSC, which leads you to think that really this is a liver gut situation. And one of the hypotheses here is the leaky gut hypothesis, that basically is that when you have increased chronic inflammation, you have increased permeability or increased movement across the intestinal mucosa. And so these things are able to move back and forth when they really shouldn't. There should be tight junctions being held together. Remember, I mentioned TGR5 earlier. This is one of the players. And so patients with PSC, if they have altered TGR5, this may also cause holes in their junctions. And then these components can cross back and forth, inducing leaky gut, which then can also promote liver disease. Next slide. So the gut-liver axis, this has really become something so, I don't want to say trendy, but it's a very popular topic right now. This is really, it is incredibly important. This is this bidirectional relationship between your gut, the microbiota, and the liver itself. And this can be, again, generated and motivated by dietary, genetic, environment factors, pollution, all kinds of things that you eat. And the interaction here really happens through this portal vein. And so if you look at the diagram below, and you see right there in the middle, the graph is not so great, but between the liver and the GI tract is the hepatic portal vein. And beside that is the bile duct. So we have blood flowing one way and we have bile flowing in the opposite direction. Well, that GI tract to liver portal vein provides direct access to the liver. And so this is why I think that over the last decade or so, we've really been focused on this gut-liver axis, on what happens when these components in the gut are released into the liver. Next slide. So we'll talk just a few minutes about bacteria and antibiotic treatments. So there have been a lot of studies that have shown that there's an overabundance of Enterococcus, Fusobacterium, and Lactobacillus in patients presenting with PSC. And Enterococcus was also found to be correlating with high serum alkaline phosphatase levels, leading researchers to think that this was potentially a target for therapy. And basically, this is a very large genus of the lactic acid bacteria, and they present as gram-positive, but they can be responsive to antibiotics like vancomycin. And in fact, vancomycin can work by altering and sort of controlling the microbiome and other immune cells within the liver. And there's been multiple studies that have shown that vancomycin and other drugs, including monocycline and others, if you give them in combination with UDCA, there have been some promising outcomes. However, this is pretty new and doesn't always work in all populations of patients. Next slide. All right, what about immune cells? We have T cells, we have NK cells, we have mast cells. Oh my, there are so many immune cells. We can't talk about them all. What are immune cells? These are just the cells that are part of your immune system that help you fight infection and other diseases. They can develop from stem cells in the bone marrow, and then they can obviously become different types of white blood cells in the circulation. Depending on which type of immune cell we're talking about, they can migrate to tissues and organs and perform the same task. Some of them mature once they get there. Some of them are recruited over and over again as if the body remembers. And so the role of immune cells within PSC is going to be really critical here, because we know that there's a ton of inflammation that's caused, and bile acids, again, can play a role also in how immune cells respond. Next slide. So what about T cells? So these are the immune cells that basically will focus on a very specific foreign particle, and they go around until they find this very specific antigen or target that they're looking for. And one of the really important things in PSC are memory T cells that they basically don't forget. So memory T cells are found both in gut and liver samples in patients that have PSC/IBD. And basically what this sort of suggests is that there's a recruitment of these memory T cells during the disease progression. And so once the memory cells are in there, they bring in other cells we'll talk about in a few minutes, like natural killer cells, et cetera. All of them have a different role that they want to play in their response to this foreign invasion or whatever target that they're looking for. There's also autoreactive cytotoxic T cells. These have been shown to be a potential biomarker and also might play a role in autoimmunity in PSC patients. Next slide. NK cells and monocytes. So NK cells are natural killer cells or natural killer T cells. They actually might have a dual role. They can either be protective or they might also contribute to damage. It just depends on the situation, the stage of the disease, and where you might find them. But here, what happens, you might see damaged cholangiocytes. Remember the cells that are lining your bile ducts, and they might actually take on an MHC class 2 phenotype. And basically what happens then is they actually become a presenting cell to recruit a T cell into the liver. So usually you have to have a very special cell to be antigen presenting, but a cholangiocyte can actually sort of morph into this antigen presenting cell, and they can recruit these T cells into the liver either to cause damage or to be protective. And it's unknown right now exactly the role that they're playing there. Is the recruitment in the beginning to potentially be protective, but then more T cells come in, more natural killer cells come in, and then you have the reverse situation where they become more damaging. And then monocytes are a white blood cell that will fight infections and can help other white blood cells remove dead or damaged tissues. They can destroy cancer cells, and they can regulate against foreign substances. And the cleaner cells is what we call them, or either macrophages or the Kupffer cells. And what we've learned over the years is that during PSC, when you have this increased inflammation, it's usually because you have a recruitment or a sort of an infiltration of macrophages who have been there to clean up, but now they're releasing other things like TNF alpha and other pro-inflammatory cytokines that can then increase the damage. Next slide. So mast cells, I threw this in because this is the work that I do, and we know that they also play a role in patients with PSC. And so these are immune cells that are primarily involved in allergic responses, but we've shown that they're increased in patients with PSC. This can correlate with also increased high histamine levels within the serum. And basically they can interact with the Kupffer cells, they can interact with T cells. Bile acids can change the way mast cells will degranulate or migrate into the liver, and they can interact with the cholangiocytes directly. So there's a crosstalk that happens between these immune cells and the resident liver cells within PSC that can drive progression of the disease. And some of the therapies here might be antihistamines or another drug called cromolyn sodium. Next slide. So one of the last topics here we'll talk about are these anti-neutrophil antibodies. So ANNA or ANCA. These ANNA is just anti-neutrophil nuclear antibody. The other one is a cytoplasmic antibody. And these are also can be found in patients with PSC, also PBC. And they might be predicative of disease progression. A study was shown that in patients that had high ANN levels were more likely to have transplant compared to the ANNA negative. And then the other, and the ANCA, these 87% of patients with PSC with or without UC, and 68% of these might have high levels of the ANCA levels, but these so far haven't become really very reliable, completely biomarkers as of yet, but there's something to keep our eye on in terms of potential biomarkers and also correlation of disease progression and status. Next slide. So research and PSC, I just wanted to throw this in here. The first article I found was back in 1952. The first case reports were presented in 1958. There were 6 of them. Unfortunately, I couldn't read any of these because there were no scripts available. But between 1958 and today, almost 7,500 articles have been published regarding the progression, targeted therapies, trying to understand the role of all these different players that I pointed out tonight with PSC. And so, we're continuing to work on this, but there's a lot of work still to be done. Next slide. So in summary, when you think about all of these, they all really come together. You have interactions between your bile acids, your microbiome, antibiotics that you might take, immune cells that are recruited. All of these are critical to the progression of the PSC, but also towards targeted therapy. We know that the crosstalk between the gut and the liver is going to be increasingly important and a major contributor to disease. If we can regulate one, will we be able to regulate the other? Multiple modes of targeted therapy are being studied, including antibiotics with the bile acid therapies. And of course, understanding how individual patients are going to respond to therapy is going to be key to managing disease progression. Next. That's it. That's all I have.
Thank you so much, Dr. Francis. And we're really looking forward to the breakout rooms where we'll get to ask you some more questions about these topics that you covered in your wonderful presentation. It's also so exciting to be here at the first of our roadmap research sessions. We have 2 more research sessions coming up, and this will be a great opportunity to continue to ask questions to our expert researchers and to each other.
Huge thank yous to Dr. Francis for a wonderful presentation, to our research experts for leading the breakout room discussions, and of course to all our attendees for being brave and asking your questions. Here are reminders about current and future programs of interest. Visit our website at pscpartners.org to learn more. First of all, please save the dates June 2nd through 5th, 2022, and plan to attend our conference in Boston. This will finally, after 3 years, be an in-person gathering combining our traditional PSC Partners Patient Caregiver Conference with the addition of an international collaborative research network component. You really won't want to miss this. Join the patient registry if you haven't already. Ideally, we would love to have every PSC patient participate to help us accelerate PSC research. Visit links to our updated COVID-19 and vaccine information. Join us in this summer's Walk the 83.01, a fun fundraiser which ends September 30th, and invite your friends and family to participate. Sign up for our mailing list and follow us on social media. Join our future Zoom rooms to get to know other patients and caregivers. We have caregivers, spouses, and parents on Tuesday, August 17th, teens ages 13 through 20, Tuesday, August 31st, and adult PSC patient ages 18 and up on Tuesday, September 21st. All Zoom rooms take place from 6 to 7:30 p.m. Mountain Time. For our Canadian participants, sign up for your mailing list and follow us on social media. Participate in Canada's Walk 83.01 also until September 30th. Please save the date Wednesday, September 29th from 6 to 7:30 Mountain Time to attend our second research webinar 201, the PSC research landscape now and into the future, identifying research questions. Dr. Ruth Ann Pye will say a few words about that session.
Thanks so much, Ricky. Tonight's Research 101 really set the groundwork for our next 2 roadmap meetings. Please join us next month on September 29th as we build upon our understanding of PSC research concepts and begin or continue asking research questions. Ahead of the Research 201, I'll also be hosting an informal Zoom room where we'll discuss tips and tricks to formulating research questions. As we know, PSC is complicated and there's a lot to learn, but our hope is that together we can suggest research questions and prioritize efforts to accelerate our path to a cure for PSC. So please keep an eye out in our monthly newsletter, social media pages, and our website for communications with more details, and we hope to see you all at the Roadmap Research 201 on September 29th. Have a great night, everyone.